This is the United Podcast Network.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you, and welcome to another episode of Voices in Validation, brought to you by the IVT Network. Traditionally, compounding pharmacists have relied on professional experience and published data to determine product stability. Based on this knowledge, compounders have extended BeyondUse dates when deemed safe and effective. With rapidly changing guidelines, these kinds of judgment calls are no longer allowable in many states, and many compounders are forced to conduct expensive and time-consuming studies to extend the BeyondUse dates for compounded sterile preparations. So today we will discuss lessons learned in performing these stability tests with our guest, Rick Rhoads. Great to have you with us today, Rick. Welcome.
Thank you, Stacey. It's great to be with you.
Absolutely. So quality control in sterile compounding is very important, and both the FDA and USP have guidelines around best practices for compounders. So let's start off with a brief regulatory review. What are the current regulations surrounding BeyondUse dates and stability testing?
Yeah, that's a good question, and it's difficult to answer actually, because it is kind of pretty complex. The way that the regulations around assigning BeyondUse date, the way that they're laid out, it's really a state by state thing. There are the national guidelines in USP, in particularly for sterile compounds in 797. But the current chapter for 797 focuses mostly on sterility assurance and not so much on chemical stability. And so there was a chapter that was finalized or was published, a revised chapter to 797 and 795, but those were appealed and then they were remanded back to the committee. So the current chapter, there's not a lot of information or guidance on what type of or how to perform stability studies or when stability studies are needed. But however, the states now, they can regulate, and they regulate on their own. And so we faced this challenge in California going back a few years ago, where they passed a regulation requiring stability studies for sterile preparations when you exceeded the default dates in 797. And that's how we got involved in it and started trying to figure out what we needed to do and also how to do it.
Sure. And so it's obviously the state boards means that each state is governed a little differently and can have different regulations. So I know, in speaking with you in the past, that obviously in California, you are performing stability testing, and that is part of the regulations out there through the state board, right? As opposed to...
Yeah, right. They are regulations the state board passed, right?
Right. So it seems to me that then diving into formal stability testing without really having a testing protocol or prior knowledge of how to, properly do this could be very challenging. And obviously, I'm sure you had to take some time to get up to speed on that yourself. What resources are available to assist compounding pharmacists in designing a study for the first time? And are you able to use stability information from ingredient manufacturers and other places like that in your study?
Yeah. So that was, I think, the challenge, because, and just to take a step back, I think one of the issues that compounders have faced is that, there's always been this kind of art and science in compounding. And, in school, you don't really learn much about the science of compounding. And so, you graduate, you have a license, and now technically you're allowed to compound, but that doesn't necessarily mean that you should be compounding, because there's so much of the science part that I think we need to be playing catch up on. And especially in just the compounding 503A compounding, there's so much that we need to learn. And, by, I think, by design or by definition, compounding is like filling in a gap. And I know this was discussed in one of your previous podcasts with Eric Costango and Kevin Hansen. I think they outlined it very well. And it's one of the issues that compounding fills in a gap. And the manufacturers have they broke off. I mean, a while ago, it wasn't long ago, and everything was compounding. I mean, all manufacturing came from pharmacist compounding, and then in the 20th century, that kind of broke off, and the manufacturers, started, I think, developing the science, which is pretty sophisticated, of how to manufacture a quality drug. And so now, as a pharmacist, we need to as we're facing these new challenges, we need to look to, in some ways, the principles of and what these lessons learned of the manufacturers and what they're doing. And so I think that's the challenge that I think we face as pharmacists is what do we need to do and how do we need to do it? There's not a clear laid out step by step. And, pharmacists like that. We like, here's step 1 through 10, go ahead and do that. That's what you need to do. And that there really isn't that. And so what we need to look at are some of the principles, I think, laid out and the CGMPs and then, how does that apply to compounding? And I think that was the challenge. So as far as some of the, references or some material that I think would help compounders. Definitely FDA guidances. There's a few guidances that are on stability studies. There's one that is still in draft form that is for 503B outsourcers and in particular the CGMPs and how they apply. There's a section that talks about stability studies and assigning BeyondUse dates. Now, I don't want to say that is what 503A should be following in particular, and because the CGMPs wouldn't fit for 503A. I mean, we wouldn't be able to fill the gap. We wouldn't be able to get the drugs out the door and to fill the gap that's in healthcare if we had to follow the strict, example of CGMPs. But they do outline some, I think, principles that we need to be following. And so that, I think, some of these FDA guidances as well. And then there's I would say most importantly is to connect with a good analytical laboratory, because they're going to be we're not chemists, right? And so we don't know how to, develop a method, and we don't know what is a good method or a bad method, how to validate it, stuff like that. So I think connecting with a good laboratory is really important. And then talking with their chemist that's experienced. And I think they can help point you in the direction, because there are relevant USP chapters on validation. There's USP chapters on, a lot of the different aspects of what you're going to have to be looking at when it comes to a stability study. There's also an article that I think is helpful that was kind of a paradigm shift for me, and looking at what is a test that would be applicable to a stability study versus the type of potency test that compounders do. And that has to do with what's called a stability indicating method. For compounders, I mean, we've been utilizing laboratories for quite a while. I mean, we send our products out. I would say most compounders send their products out to laboratories and have them tested. The difference is that generally they use what we call a release method, which is just like a general method that would cover a certain drug, but it's not developed specifically to your formula. And so it may not be accurate when determining stability. So there's kind of a paradigm shift that is needed to look at the importance of developing a stability indicating method. And that's where things get a little bit tricky. But the article was published by USP, and it was written by Lloyd Allen, Gus Pisani, and Alan Parr. There was another person on there as well. I can't remember the name. And let me see if I can get the
Yeah,
the
we can try to find that and include it in the show notes, a link in the show notes. So folks.
Yeah, a link would be good because I think it's helpful. It's written by pharmacists, and I think it's for pharmacists. It's very helpful in that. So those are some of the good references that I think once you start there, then it's going to just open up, and there's a lot more information that that pharmacists need to learn before doing the study.
Absolutely. So thanks for including some of those references and resources for our listeners, because I think because there is so little by way of guidance on this available, it's really important to have a starting point, of where you can even go to get some direction and begin to put together your stability testing plan, right?
Yeah.
I know you talked about challenges in terms of finding a good laboratory to connect with and making sure that you understood what the USP and state board guidelines are in California. Can you talk a little bit more or describe some of your own challenges in preparing for and conducting the actual studies, the stability studies?
Yeah, absolutely. I think that's really the reason that I wrote that article is because we face quite a few challenges. I want to say something about the last question though, real quick to wrap that up, because you asked kind of another question about, using, I believe you asked something about using studies from other.
Yeah, or information that's published, right, from the ingredient manufacturers. If you are able to get information, is that usable or not usable?
Yeah, and that's a really important point, because right now the chapter allows for kind of general stability study. If you're able to, you can use data from resources that are just general stability resources. But I do believe that you can use going to specific stability studies, using stability indicating methods, that you can use data from other sources, including ingredient manufacturer or ingredient repackagers. And USP actually has compounding monographs, and they publish with their stability indicating data. You can use their extended dates. So I think that's pretty accepted practice, as long as it's the same formula. So it's got to be identical in ingredients. And general essential compounding steps, as long as that fits, and the same the same package as well. And I think the container closure, as long as those match, then you should be able to use it. When they differ, I think that's when it becomes a problem, when it's not the same formula, but now you're applying that stability information to a new formula, then it doesn't fit. But going back to the recent question about challenges, again, this is something that's new for a lot of compounders. It was new for us. I had never designed a study, or carried one through. So I think there's a few things that I think were challenging. The first part was just the design, because it takes a lot of time and effort to determine, what are the different data points, what points am I going to be testing, what days, and what tests am I going to be performing at those different points. Once you develop this method and validate it, you're kind of set in stone, you're locked in. So you have to look at all the different variables that go into making this formula, and you have to control for those variables, because if something changes and you started the study and you're 60 days in and you realize, oops, I made a mistake, that I used the wrong excipient, or actually I used a different concentration, and then that becomes a problem, and you have to restart the study. And we actually encountered that, where we ran into a problem with one of our starting ingredients, an aliquot that was made, and realized that although it was within the accepted limits, it wasn't a tight enough range that we wanted. And so we had to go back and redo it. And that was, expensive, and it set us back a few months. And so I think the design of it is important. And then also carrying out just the practical part of, you have to create a study drug to send out. And that's not something that we typically do. And so, you have technicians. I mean, usually in pharmacies, you have pharmacy technicians that they're really the proficient ones when it comes to sterile compounding or, in compounding in general. And so you have to relay all the details of this study and make sure that they're communicated properly to the people that are going to be doing the work. And if they're not, small things, just practical points about, the shipping conditions and who you're going to be using to ship the study drug. Is the laboratory ready to accept the study drug? Those small things may seem like, okay, well, they're not really that important, but when you have everything on the line and the laboratory is ready to start the study and you mess up one of those things, again, now you have to restart. So some of the practical aspects of communicating all of that to the staff that's going to be preparing the drug, those were some of the challenges that we ran into. We just weren't really prepared for and controlling for some of those variables.
Sure. And so I imagine, obviously, you're here to tell the story, so you made it through this process, but it does sound like there were a lot of little gotchas that you wouldn't necessarily have thought of until you were right in the midst of trying to put the study together, right? Have you found that stability testing has gotten easier, following your initial study, now that you've been doing it for a couple of years at this point?
Yeah. It is definitely, there's a learning curve initially, and then you start to figure out some of those pitfalls and try to avoid the problems moving forward. So yeah, it definitely gets easier. And we have been, it's probably not a process that you just do one time. A lot of times you'll do it for other drugs, other strengths. You may make changes. And so it's kind of an iterative process. We're going to continue to do it. And it does get easier over time. But it's still a challenge. This is not something that we do. We don't usually employ the personnel to do this like a lot of the manufacturers do. They have people that are experts in a lot of the various disciplines needed. And, for community compounders, hospital compounders, you have management, you have pharmacists, and you have technicians usually, maybe a few others. So, it definitely is always a challenge when you're going to be doing that, especially when you're spending the type of money that, is needed to, and the time that it takes to do it. So it is always a challenge.
Depending on the size of your hospital or facility and the number of staff you have on, it can be even more challenging. The smaller you are, the busier you are. Those all play factors, or are factors really, in getting this done the right way and not having to restart so many times. So just a couple questions, follow-up questions. Do studies need to be conducted for every compound, that's regularly prepared for single patient use? And what about batch compounding? Can you talk about those two things and how they relate to the stability testing or the expectations around doing these stability tests?
Yeah, generally it's irrespective to whether they're patient specific or whether you're making a batch. For 503A, everything is patient specific, but you may be making kind of a one-off thing where you're making one drug for one patient. And it usually would not apply. It would not have an effect regardless of whether you're going to be doing that. But from a business standpoint and a practical standpoint, so if you have fast-moving drugs that you're making big batches for, it usually is more practical to have a longer date. And so some of those studies are more likely to be done on, the ones where you're preparing bigger batches for.
That makes total sense. If it's just a one-off drug that you're making once and it's being used pretty quickly, I would imagine that that's not as big of a deal, and not necessitating a stability test. So what are the differences in designing studies or in carrying out the studies actually for low versus medium risk compounded sterile preparations? And you can talk to that a little bit. Does it matter, what the risk factors are, I guess, as to how you design and carry out these types of studies?
Yeah, so the current chapter designates risk levels based on sterility assurance. So it's not necessarily based on the formula or the drug or the date, but it's based on the sterility assurance levels. And there's a lot of different factors that go into that. You know, how many different ingredients that you're using, in some cases, what environment that you're compounding in. And so that would all be in terms of sterility assurance. And that would, again, be separate from the chemical or physical stability part of it. So, yeah, they would be pretty much separate with from the different risk levels of what you're going to be doing. You will be doing some sterility testing and usually looking at preservatives and such. The one thing I will say, though, when it comes to that is I do think when it comes to assigning the date and utilizing the data that you have and now assigning a date, you're probably going to be more conservative on riskier products, right? Because the date is not just, you know, what the chemical, physical stability and compatibility is. It's also what's the risk to my patient over a period of time. And the higher risk drugs have a higher risk, obviously. That's why they named it that way. And so you may not want to put a long date on that because there's a greater risk to it. And that's where a lot of the process controls and things that would be like facility controls that go into place to make sure that you can make the study drug and that study drug is good for so long, but you made it on one day. The question is, can you make it every single day, make it the same way and be accurate in that and produce a drug that's going to be safe? And that's usually why BeyondUse dates are quite a bit more conservative than expiration dates, where they may have 1 year, 2 years on it. They were made under CGMP conditions with a more robust quality assurance program, and so that's why the dating for sterile preparations are much more conservative than manufactured products. Manufactured products.
Right. Okay, and that makes sense, and it makes total sense. So, what have you learned in your facility about ingredient quality, and in particular bulk APIs, container integrity? I noticed. I know you said one of the things you had to consider was if it's the same container or same packaging, and dosage strengths on the impact of product degradation. I got it.
Yeah.
And stability. So, I mean, I'm just wondering, like, I get that stability and shelf life is different than data integrity or product quality, but I feel like they all are interrelated a little bit somehow. So I'd love to get your thoughts on that or lessons learned here.
Yeah, I mean, your drug is only as good as your starting ingredients, right? I mean, if you have problems with, the starting material, then, you're not going to have a good study, right? So it's important to have some qualifications around and some validations around what you're using, not only for the study drug, but also just in operations in general. And, this has kind of been, a hot topic actually recently. The FDA's had some actions against some repackagers, and that I think opens up a whole new can of worms of what type of qualifications and quality agreements need to be in place with compounders and the repackagers, because usually compounders really trust those repackagers quite a bit. They are probably registered with the FDA, licensed with the State Board of Pharmacy, and then they source those APIs from other manufacturers around the world. And there may be dozens and dozens of manufacturers, and they all get funneled through the repackagers and then come through to us. When we see the product and we're using that product, it's labeled with the repackager's name, not with the original manufacturer's name. So that may change. The manufacturer may change from lot to lot. And so that's something that we have to take into account. I think this is going to be worked out over time. you know, in the near future, we're going to have to figure out what that looks like. I think for aseptic for manufacturers in general, they have to perform a lot of their own in-house testing. And compounders generally don't do as much testing of the incoming starting materials, incoming ingredients that they get. They rely a lot on the repackagers. And we'll see how that changes, whether that changes at all moving forward, or whether we just need to use quality agreements with the repackagers, because those variations do exist, as well as the container closure, because that can have an effect. There was an issue a few years ago with some syringes that compounders were using, and the manufacturer of those syringes changed the component, one of the components. And it started having an effect. They started seeing subpotent compounds, and they, all of a sudden this came up, and they didn't know why that was the case. And as it turned out, the manufacturer of the syringe said, yeah, well, this was changed, and we never really intended for the syringe to be used for storing drugs for a period of time. So that kind of opened up, again, another challenge that compounders have to deal with is how do we control for some of those variables? And it is one of those issues. And I point out in the article that, if you're going to be making any intermediate ingredients like aliquots, a lot of times pharmacists will make an aliquot of a certain ingredient that will be used in several different formulas. Mm-hmm. And if you're going to be making that, I think important, and we found this from the study, to have some parameters around what's acceptable, what kind of attributes you need for that aliquot, instead of just making it up and assuming that it's going to meet the quality standard that you have, because at 93% potency is going to behave much different in a compound than a starting material of 103% or 105%. So trying to narrow that down to a little bit more of a narrow range for the aliquot. So starting ingredients are important, and I think it's an important thing that kind of came up for us in the stability study that we did. And I think with the recent news that we've seen and the action taken by the FDA, we'll see how that works itself out. But compounders are going to be more vigilant in the future of the ingredient, the attributes of the quality of the ingredient, and then how they qualify those different repackagers and what regulations exist around communication of the original manufacturer of those ingredients, and a lot of the different issues around quality of that. So it's an interesting topic, and we'll see how that shakes out in the near future.
Yeah, no, for sure. And it sounds like, you weren't just putting a test together here, a stability test, designing that for one-off. You're really building your protocols, right? As a facility, yeah, as an organization, to make sure that every time you perform these types of tests, you're doing it the same way, and that everybody knows you start with the same, base product, and all of the things all the variables are kind of put in line so that you can reproduce what you're doing over and over with consistency, and not have to worry as much about the fluctuations.
Yeah, absolutely. Yeah, I think that's the thing, just to wrap up your last thing. I think once you have a study, now you have to implement this into your daily operations. It needs to not just be assigning a date, but there's some specific things that are now kind of set in stone with what you studied, and now that needs to be part of your daily operations as well. You're
exactly.
Right.
And it's reminded me of, and you mentioned it, but good manufacturing practices, right? Like that's what they do. I mean, it isn't just about producing a good product every time, but it's about making sure that all of your procedures and policies and whatever are in place, so that it doesn't happen by accident, right? It happens every single time.
Right.
And that's the important, I guess that's one of the important takeaways here.
It should be designed. Exactly.
Yeah, for sure. So, in your experience, Rick, during a study, what is the most likely source of error? And how can our listeners avoid, these types of mistakes?
Yeah, I mean, I think I talked about a couple of them. And I can't speak to the lab side because there are errors that occur. I think if you were to speak, to professionals on the analytical side, that's a difficult science, and errors can occur on that side as well. But as far as planning the study, I think just not really controlling for the variables, not thinking through the design of the study properly. That is one of the biggest sources of a problem. There are a lot of different unknowns. I mean, you don't know what you don't know. And so initially, you can't anticipate some of the issues that will pop up. So I would say study design. That would be a pretty big source of errors that can occur. And then the last thing would be what I was talking about earlier about, okay, now I have this data. How am I going to take this data and integrate that into and make that part of my practice in pharmacy? How do I implement that into my in my training and my SOPs and my formulas? I think that's another piece that is easily overlooked and can become a problem after the study. Sure.
Thank you. I'm just thinking about the we're still here 11 months later dealing with the effects of the COVID pandemic. Starting early on, there were global drug supply chain was definitely impacted in a lot of different ways. The drug supply chain, the PPE supply chain, like on and on, leading to many supply disruptions and shortages of drug products. And I'm just curious if you've seen any sort of impact on the assignment of BeyondUse dates or stability testing requirements as a result of drugs not being available or substitutions needing to be made.
Yeah, there's a couple of things that I think have become an issue over this last year. Certainly sourcing products has become a problem. Sourcing the API, the active pharmaceutical ingredient, I mean, that's certainly an issue. We've seen a disruption in the supply. And obviously PPE is an issue. And I would say that's probably the biggest problem for compounders is, accessing, everything is built, this whole, all of the BeyondUse dates are built with the assumption that there's a certain amount of sterility assurance that's in place, and that sterility assurance is based, a lot of it is based on, not only the facility, but also the protective equipment that we have, because we have basically all we're doing, it's a manual process. And so we're relying on people to go in and do this compounding. Well, we're the biggest generator of particles in that environment. And if you don't have the proper PPE, that's a big problem. And that would affect the BeyondUse dates. USP did publish some guidance on that, operational considerations when compounding. If you're unable to access and source the proper PPE, I mean, people still need their drugs, right? And there's a lot of critical compounding that's done, and some of these hospital compounders and even community compounders can't just tell people, oh, I'm sorry, I'm not going to give you your antibiotic. I mean, it needs to get out the door, and patients still need to be treated. So I think some creative ways were I think come up with this article, USP article, or considerations that they published, and a lot of that had to do with shortening BeyondUse dates, and if you have to reuse PPE or if you have to go without certain PPE, then a shorter date is necessary, because the longer the date that you have on the product, the greater the risk it is to the patient, because, bacteria, you may have a small amount, or you may have, some contamination. You don't want any contamination, but if you have some contamination, it's going to be a bigger problem for the patient 60, 90 days from now, 120 days from now than it is on day one. And so what was basically expressed in this was, minimizing the BeyondUse dates. And there were some creative ways to to help with the shortages in PPE, but it really is an issue that I've seen come up in compounding is the drug supply chain because of COVID.
Yeah, and hopefully we're on the downside of the supply chain issues in terms of PPE.
Yeah.
But certainly, I think we're going to continue to see shortages of APIs because of just maybe the locations we have to source them from, geographically speaking, and what's happening there. And of course, there's new guidances or guidelines supposedly that will be emerging from the FDA on where certain things can come from that we're going to use here in the US. And that's a whole nother story. But I mean, those are things that you obviously in the business are going to have to keep your eye on so that you can adjust your compounding and your testing accordingly, I would think. So you've been through this in terms of designing and implementing stability testing, doing your studies. What is the time frame for conducting a study like this? How long did it take you guys from beginning to end, so to speak?
Yeah, it does take a while, unfortunately. And the main reason for that, I mean, if you have the study itself is going to have to take, whatever the time period it is that you want, whatever dating you want. But before that, you have to design the study, and then you have to design the methods that are going to be used for the study. And that gets into the analytical chemistry side of it. Again, not something that pharmacists typically deal with, but now we have to start learning about it a little bit because it matters. And those methods take time to develop and validate. And so there's a whole validation process that takes place. And that's, I mean, months and several months to even get to a point where you're ready to start the study. So if you're just getting started, I would say 6 months to a year, probably by the time you're completed. Because you could, maybe if you get lucky, you can go faster than that. But just from our experience, I mean, it took a while to get to a point where, the lab has to devote quite a few resources to the project. There's a lot in terms of coming up with the exact, methods for each of the APIs that are in that drug, in the formula. And then the whole validation that takes place to make sure that they can do that properly. And so that takes a while, several months usually, just to do the entire validation process and to start the study itself. So, probably depending on what your date is or what your dating that you're shooting for, 6 months to a year, probably.
That's great. I mean, that's great for our listeners to know, right? So you got to plan accordingly. It is a lengthy process. You have to really buy in to the whole process of it and be committed to following it through, because it's certainly not something that you're going to do, in a matter of days or even weeks. Sounds like. Right.
Yes. Correct.
Well, Rick, some great information today. I'm going to also share the article that you wrote with our listeners too in the show notes, and I'll see if I can find some of the USP guidances that we can share as well. And if there's any other, articles or links out there on the web, we'll definitely share those as well. But before we wrap up today, I just wanted to get in closing, give you an opportunity to share any tips or advice that you'd like to leave our listeners with, now that you've gone through this, and it's kind of been more than a year, I guess, since you've completed your initial study and you've been working with this. So what kind of advice or tips do you have for our listeners?
Well, I would say that it's worth exploring, especially for your products that you have longer dating on, and products that or preparations that are being dispensed in high volume. Ultimately, it's not just about what the standard is. It's not just about what we have to do, although I get that, and I operate in that space as well. I understand there are expectations, and we have to meet those expectations, and to a large part, they dictate what we do. I would also say that, as pharmacists, it's important for us to realize that, the burden of the quality of the drug and the outcome of the patient does in a large part fall back on us. And so we need to know not just what we have to do, but we also need to know what we should be doing and what is the right thing to do. Sometimes it may not be achievable. Again, I see the GMPs as a guiding principle. We may not be able to do the GMPs, but if we're having trouble understanding a challenge and how to overcome that challenge, the GMPs may be a good source for us. So I would just say, for any compounder, look at what you're doing, and if you feel that it's the right thing to do, I would encourage you to look into it, to explore it. It is a significant investment. So, I don't think I can just easily tell people, go ahead and do it. It's not that simple, and it's not cheap. But again, if you can do it, and especially if you're putting longer dating on there, I think it's something that you should look into. Connect with a good laboratory, one that is experienced, that's done this before, and start the conversation. I think it's worth doing, because when a patient is using our preparation that we dispense 90 days, 120 days, or whatever date we put on there, 60 days out, we want to have some assurance that it is what we say that it is. And it does meet the quality standards that we want for not only on the day that we dispense it, but also at the very end of their therapy. So it was an important piece that we found helpful in our pharmacy, and I think other pharmacies may find helpful as well.
Yeah, and it all comes down to patient safety, right? You want to make sure, as you just said, that the drug is being delivered the way you intended. And it's the efficacy is the same as it was when you made it, even if it's 2 months, 3 months down the road. So very important.
Yep.
Thank you, Rick Rhoads. Great to have you here with us. I really appreciate you being our guest this week.
Thank you very much. It was great to be on.
Of course. Also, I want to give a big shout out to our producer, Ben Kitchen, and the Studio 21 Podcast Cafe. And lastly, a big thank you to you, our valued listeners. If you have topic requests or guest suggestions, you can message us via LinkedIn or through our website. And if you enjoyed today's episode, head on over to your podcast player of choice and subscribe, rate, and leave a review. Plus, don't forget to share with your friends, colleagues, and online networks. We really do appreciate your help in spreading the word. For today's show notes and resources from this episode, please visit www.ivtnetwork.com. We'll be back again next week with another great discussion. Until then, make it a great week.
The views and opinions expressed by the hosts, guests, or callers of this program do not necessarily reflect the opinions of the Studio 21 Podcast Cafe, the United Podcast Network, its partners, or affiliates.